Cameron Fanup, and I am the Director of Marketing and Sales here at Bioservices. I will be serving as the moderator for today's webcast. Bioservices has been developing its proprietary electrospinning technology for almost 20 years. Our Bio-Spun materials are currently being used for many different applications, ranging from medical devices and benchtop research tools to consumer products such as skincare applications. Biosurfaces typically works in two ways. First is to work with partners to apply our technology to the partner's applications, and the second way is to apply our biospun materials to develop technology for its own applications. For the biospun cell chamber, this device bridges both of these areas. In 2017, Biosurfaces was approached by Takeda Pharmaceuticals and asked with making three devices for gastrointestinal or GI applications. The Bio-Spun cell chamber was one of those devices that was generated from this program. While Takeda had interest in the cell chamber for GI applications, there are many uses for this technology outside of that space. Many of those applications are now being pursued by both Biosurfaces and our partners. In today's webcast, you will learn more about how the Bio-Spun cell chamber came to reality, what pitfalls were hit, and how the company overcame them, and why this technology could be a game changer. We have assembled a group of people responsible for bringing this vision from concept to reality. That panelist today consists of Biosurface's very own Mr. Matthew Fanup, co-founder, president, and chief technology officer, Rayan Kasab, the director of device development, and Dr. Joshua Chakravarti, senior research associate. First question is for Matt. Can you tell the audience what the Bio-Spun cell chamber is?
Absolutely. The Bio-Spun cell chamber, in the simplest of terms, is a pouch made from our proprietary electrospun materials into which different cell types can be injected into. The chamber is then sealed using ultrasonic welding to keep the cells in and the body cells from getting into the pouch. The cell chamber can then either be used as an implant into different parts of the body, depending on the application, or can be used for benchtop studies.
How does it work?
The Bio-Spun cell chamber acts sort of as a factory in that the cells inside the chamber use nutrients from outside the chamber that diffuse through the wall to keep the cells nourished. These nourished cells inside the chamber can then be used to produce a target drug such as an antibody if the cell was previously genetically altered prior to enclosure, or produce a protein that it would normally produce such as insulin. The chamber serves to protect the enclosed cells from being attacked by the body's immune system, since the cells in the chamber would be considered as a foreign invader. The outer electrospun materials provide a surface for the body's cells to grow into and heal.
How did this project first start, and what data has been shown to show if this product could even work?
We were first approached by Takeda Pharmaceuticals several years ago to see if Biosurfaces could develop a prototype cell chamber that could be used to deliver genetically modified cells that would produce a target drug to treat gastrointestinal disease. Our team underwent several iterations of this prototype device before landing on the current device that is being evaluated by different customers today, all of which are outside the gastrointestinal space. A few key metrics that we evaluated to show that the device was working well was cell viability or health over an extended period of time, target drug release, and device healing.
What benefits does Biosurface's cell chamber have?
Our Bio-Spun cell chamber is flexible, can be formed into different sizes and shapes if necessary, can accommodate different cell types, can maintain cell viability or health. It can protect the enclosed cells from immune attack, and does not undergo scarring, also known as fibrosis. Cell chambers produced by our competitors have had issues with achieving one or more of these benefits.
Thank you, Matt. Next question is for Rayan. How did you start on the project, and what is your current role on the development of the product?
I started working on this project when I started working at Biosurfaces, which was over 4 years ago. At first, it was just another project that I was working on because I didn't really know much about cell chambers in general. It was just very new to me. But as time went on and as I learned more about it and the need for a device like this, because there really isn't much out there, my interest grew and I became invested in it. So it became my biggest project that I dedicated most of my time to. My current role is the lead on the development of the cell chamber along with the next generations of this device. For example, I'm working on on developing a chamber that has more surface area on the inside of it to maximize the number of cells it can fit, and also developing a resealable port that will allow for multiple cell injections without the need of any equipment to seal off the hole that was left by the needle.
What were some of the pitfalls you have come across?
One thing that was challenging that we spent a lot of time working on is designing the walls of the cell chamber in a way that will allow the product that is being produced by the cells inside the chamber to come out, but at the same time not allowing anything from the outside world to come in so we can keep those cells protected. Finding the balance was a little bit difficult. And another challenge was developing a subdural cell chamber that was being tested inside a rat's brain. Right now, I'm making these chambers by hand, so I'm sure you can imagine the challenges that come with making a device that's only about 1.5 millimeter in diameter, which is kind of like the tip of a sharpened pencil, kind of that size.
How did you overcome these issues?
Through a lot of trial and error. Like, for example, in regards to the challenge that I talked about with designing the walls of the cell chamber, we designed multiple iterations of it and tested each one in the lab until we found the one that gave us the best results that we were looking for. It definitely takes a lot of patience, but it's worth it in the end.
What are you looking forward to in the future with this product, and where would you like to see it go?
I'd like to see the cell chamber implanted. I think the ultimate goal is to help people. So seeing this device become a success and help treat different diseases is what I'm really looking forward to the most.
Awesome. Thank you, Rand. Next questions are for Joshry. Joshry, what is your role on developing the cell chamber?
As a researcher in the R&D group, my role is to test the biocompatibility of the device. I help to develop protocols to evaluate the safety and efficacy of the device and develop standardized procedures to look into the evaluation of the device.
What did you learn from first working with this device?
The Bio-Spun cell chamber are very easy to handle and load cells, and the dimensions of the cell chamber can be customized depending on the application to support growth of several millions of therapeutic cells inside the chamber. And the overall device geometry determines the cell content and thereby the rate of therapeutic molecules that are being released from the device. Also, it is very important to achieve a high cellular density inside the device in order to produce therapeutic levels of the drug. So some initial proof of concept studies that we did was to quantitate the number of cells surviving in the device and optimize the initial cell loading conditions, which could lead to high density cell survival. And from these in vitro studies, we learned that the initial cell load is a critical parameter to achieve long-term implant survival and efficacy. And we also characterized therapeutic protein release from the devices in vitro for over 65 days. And subdural and subcutaneous implantation studies in mice, we saw that the chamber could maintain cell viability and allow diffusion of therapeutic drug through the membrane for over 50 days. And we were very excited to learn that the device could protect the engineered cells against the immunological response and had excellent tissue incorporation in the animal with no evidence of fibrosis surrounding the device.
Where have these initial studies led you in relation to developing the next generation device?
Currently, we're looking into loading cells into a newer device that is designed to have more surface area for the cells to grow. And we are looking at different cell dosage that can be put into the device to maximize the loading capacity of the chambers to exert a practical therapeutic function. And in order to advance the Bio-Spun cell chambers towards clinical application and transfer this technology to humans, the next step is to scale up and evaluate cell viability and biocompatibility of a bigger size cell chamber device that can be implanted into humans. And we are also looking into ways to freeze cell-loaded Bio-Spun chambers. And the goal of this study is to look into the aspects of cryopreservation that is directly relevant to clinical situation and to have a stable and secure storage of cells in the device. This would provide us with the capability of making large quantities of device available for subsequent use and include flexibility and predictability of delivery for clinical scheduling and global availability with transport not being time critical.
Thank you, Joshua. The last couple of questions are for Matt. Can you talk a little bit about the history of making cell chambers?
There have been many companies that have attempted or are currently evaluating cell chambers, or what they call macro devices, for delivery of their target cells into areas such as the eye, below the skin, or subcutaneously, or into the abdomen. Many of these companies put so much effort into cell development that they forgot about how they were going to deliver them. Many cell lines have died on the vine because there is no good way to deliver them. I sort of equate this to having a Ferrari but with no engine. Nice to look at, but it's not going to go anywhere. There are several companies that have bypassed the idea of protecting the cells from the host immune system by using drugs known as immunosuppressants given to the patient to prevent their immune systems from attacking foreign cells.
What has historically been issues with other cell chamber devices when they were implanted?
As the panelists have talked about, and I mentioned earlier, fibrosis or scarring of the device has been one of the bigger issues that you can come across with other types of cell chambers. Scarring basically is equivalent to a person getting a splinter in their body. As you know, the body walls it off so it's isolated. Now that gives you that hard feeling on the skin. The same thing happens with the cell chambers that are developed by other groups. This walling off occurs around their cell chamber, which prevents nutrients from getting into the enclosed cells and also prevents the therapeutic agent from getting out, basically rendering the device useless. And this whole process actually occurs within 30 to 45 days. Other issues are preventing the immune system from attacking the cells inside, as Rayanne had pointed out in our initial iterations that we looked at, and also creating a device that can continue to generate a therapeutic delivery concentration over a long period of time.
What are the future applications for this technology, and why are you so excited about its potential?
We are really excited about the possibilities for our Bio-Spun cell chamber. We have several groups that are currently evaluating this technology in preclinical models, as Joshua pointed out and Rand pointed out, and they are focused on treating things such as diabetes and liver disease. Additionally, we have other groups that are looking at the cell chamber to deliver genetically modified cells that have a biotherapeutic agent that they are producing. The initial results that we've seen from all of our partners has been really encouraging, and we look forward to working with them to bring this technology to patients one day. I think that's a goal of all of our members of our team, is to make a difference in the life of the patient. We look forward to working with different partners that are interested in delivering their cells. And, I believe that, this type of webcast can serve as an invitation to any potential other partners to open some discussions with us to try to move this process forward for their cell applications.
I would like to thank all the panelists for taking time to speak today, and would also like to thank you for taking the time to listen to this webcast. If you are interested in learning more about the Bio-Spun cell chamber technology, or would like to make a suggestion about a future webcast topic, please feel free to reach out to us at info@biosurfaces.us. To learn more about Biosurfaces, please visit our website at www.biosurfaces.us. We'd love to hear from you. Until next time.